Hello, I'm Sandra. Thanks for being here. It's the poster about my master's project named Uncovering the Biogeographic History of Neotropical Tillandsia, subgenus Tillandsia. Well, for taxonomic context, we have the order Poales, in here the family Bromeliaceae, which is the family of the pineapple. The subfamily Tillandsiidae distributed from the southern of United States to the southern of South America. In here, the genus Tillandsia have about 717 species into 7 subgenera. The subgenus Tillandsia is the most diverse and have the well-supported clade and is divided into K1 and K2. Some key innovations that can explain the large distribution are epiphytic or epiphytic lifestyle, specialized trichomes for the absorption of water and nutrients, cause the roots were modified just for the hungering, bird pollination, seeds with plumose appendage, atmospheric rosettes with tank metabolism, or species with central tank and seed-free metabolism. About the biogeography, Bromeliaceae have arisen in the Guiana Shield about 102 million of years ago, and from here had a centrifugal dispersal. One of the lineages correspond to Tillandsioideae with a diversification about 37 million of years ago. From the northern of Andean zone, we have the subgenus Tillandsia with a diversification about 18 million of years ago, and two dispersal routes, one through the Central America to Mexico and USA, and the second through the central the Andean zone to the rest of the Neotropical. And we know the blue area is the diversification center for the subgenus Tillandsia, but also we know it. So the aims were obtain complete plastoms from off-target high-seq and shallow sequencing for a representative sampling of Tillandsia subgenus Tillandsia and selected outgroups. And third, define scaled phylogenetic relationships in the subgenus Tillandsia and perform a reconstruction of the biogeographic history of the subgenus Tillandsia. Well, all with the following method. First, we meaning plastic information from the off-target and genome skinning and did the assembly of complete chloroplast genome. And finally, the plastome annotation. In the second part, we did the alignment and obtained the phylogenetic reconstruction. With that information, in the third part, we obtained geographic records, we proposed ancestral areas, and we test several biogeographical model tests. So in the results, we have 5 areas: the blue, Central America, Mexico, and southern of USA, Caribbean zone, Andean zone, Amazonia, and Atlantic forest in Chaco and Dominion. Our phylogeny, we have it's well supported. We have the genus Tillandsia, like no monophyletic, and we have in here the ancestral node for this genus Tillandsia with 86% of probability to be distributed in the Andean zone and just 14% to be distributed in the Andean, but also in Caribbean and Amazonia. In the subgenus Tillandsia, we have 66% of probability to be distributed in Andean, but also in Central America and Mexico zone. 17% to be distributed just in the blue area, and 15% to be distributed in the blue area, purple area, and Caribbean zone. Zooming in subgenus Tillandsia, we have in here the subgenus. We have in here 3 main lineages. One with 47 probability to be distributed in blue area, Andean zone, and Caribbean zone. 36% to be distributed in Central America and Mexico and Andean, and 15% of probability to be distributed in the blue area, but also in the Caribbean zone. The second lineage have 100% to be distributed just in the Andean zone, and all the species are distributed just in that part. Finally, in the clay K, we recover the K, K1, and K2, the three with 100 of probability to be distributed in Central America, Mexico, and the south of USA. But in the case of the K2, we find a species which expanded his range distribution from the blue area to Caribbean zone, from here to Andean zone, from here to Caribbean and Andean, and finally from here to Caribbean, Andean, but also in the Amazonia. So we conclude that we obtain a strongly phylogenetic reconstruction in Tillandsia subgenus Tillandsia because of our sequencing strategy. In the subgenus Tillandsia, we recovered 3 main lineages with high support, one in Central America, Andes and Caribbean, the second just in the Andean, and the third just in the blue area. But in the case of the clay K2, well, that contain species which expanded their range distribution to Caribbean, Andean, and Amazonia. Finally, we want to say thanks for the attending.